Lights on, everyone. Plugged in the lecture lights on.
Oh, okay.
Yeah.
The watch.
Okay.
Great.
So that Eugene, so I have to talk about that. So you just stand next to me. All right. Let's get started, everyone. So it's my pleasure today to introduce Allie Young. Alana's actual advisor is Tom Miller, who couldn't join us in person today because he's out on St. George Island counting plants. I believe he's with us on Zoom though. Is that right? Yes. So Tom's a plant biologist, and Elena is not the first marine biologist to come through Tom Miller's plant biology lab. So I think that says something about Tom really wanting to be a marine biologist instead, actually. So let's see. Elena started with us as a master's student in the Ecology and Evolution program in 2023. And when she got here, she was in Dan Okamoto's lab. And unfortunately for us, Dan Okamoto left in the first semester of Elena being here. So she then moved to Tara Stewart-Merrill's lab and to Tom Miller's lab. And unfortunately for us again, and for Elena, Tara left us. So she has, but Tara, Dan, and Dean Grubbs, and Tom Miller, and myself are still supporting Elena, even though she's had some fluctuations in her team. So before her master's, Elena was actually an undergraduate here in biology at FSU. She got her undergraduate here in biology. She interned with Kim Hughes for a while. She worked at the Gulf Specimens Marine Lab. And so every time I see Elena around, she's got a million, it seems, students around in the lab. So she's either corralling lots and lots of students. I counted 21 DIS students in 2 years, and it's probably more than that for all sorts of other types of students as well. And if it's not that, I see her at the Marine Lab with her head buried in a shark carcass, dissecting out some squishy bit. So every time I see her, she's always working incredibly hard. So it's going to be really fun to see what she's done with all that busyness. So Elena, take it away.
Thank you. Awesome. Thanks, Scott, for an awesome introduction. Hi, guys. Today, I'm going to be talking about my thesis work, which is looking at parasite infections in marine species. So specifically, we'll be looking at patterns of tapeworm infections in blacktip sharks. Before I break down what each chapter is going to cover, let's go over some background on the system. Host parasite systems are definitely intricate sometimes, so that ecology is essential to understanding. So starting very rudimentary, what is community ecology? I know many of you know and have probably seen this diagram or something similar to it before, but just in case, ecology happens on multiple levels. The lowest is going to be that individual level where we're focused on the organism itself. Above that is population biology, which is looking at one population of one species, but multiple individuals there. And then community ecology, we are looking at the interactions that are taking place between different species. You can keep going on up. Ecosystem ecology, for example, pulls in those abiotic components, but I think that's enough for us since we're just working on community. So something really important about community ecology, or something that would help you do well, is understanding species distribution. Since we're trying to understand who's interacting with each other and where they show up, you need to know how to collect data on species distributions. And so what I mean by this is species will have a fundamental distribution, which is the amount of space that they can take up, maybe because of a environmental tolerance range. So for example, on this environmental gradient on the x-axis, let's imagine that that's temperature. And so when you are on the left side of the axis near 0, those are going to be cold temperatures. Other side's going to be very hot. So if bell curve A represents species A, this species has a low environmental tolerance, low temperature tolerance. So it can be in cold environments, but we see that about halfway through the axis, it no longer can survive in those temperatures, but it overlaps with B. So we know where host A can show up, and we know who host A is going to interact with. And so this is going to help us understand patterns of biodiversity and what keeps our ecosystems functioning. And these principles do not just apply to free living species, they also extend to symbiotic relationships, such as those that take place between hosts and parasites. And this is a great opportunity to look at community ecology because obviously if it's a host and a parasite, we know we have two different species that are interacting with each other. You don't have to go look for those interactions taking place out in the environment. And tapeworms are very helpful for doing this kind of study because they are in just about all organisms for the most part. You'll find them in the intestines of their definitive host. And so that means they're going to be localized in this small region as well. So they're easy to census and ensure that you've accounted for all individuals in the community. And so that is a level of completeness in data that we don't usually see in community ecology. Let me give you a little bit of background regarding the tapeworm life cycle. That's definitely going to be helpful for understanding things moving forward. We're going to start up in the top corner, proglottids. Proglottids, if you're not familiar, are the segments on the worm, and these are essentially egg packets. And so a mature worm, like what's pictured, is going to drop these segments out into the environment. And those are going to be in the water column or on the ocean floor, where demersal species and filter feeders are going to come through and pick up those eggs. Once they have consumed those eggs, larvae usually will encyst in the muscle of that intermediate host. And then at that point, this tapeworm is essentially waiting for that host to be eaten by another host. And so that's going to be called our definitive host. And that's the host that the tapeworm is trying to get to because that's where it can complete its life cycle and become reproductively mature so that it can start that cycle all over again. And so we know the definitive host that we're going to be working with is the blacktip shark, approximately what they look like. And so they are a great predator for looking at this system because they link multiple habitats. They have very fine scale movement that among their home ranges. So many times we'll find juveniles in nursery areas that they'll revisit year after year. And adults will move offshore because they're migratory. And something to consider there is that we also, when they're migrating, see some sexual segregation. So our male and female sharks are not migrating together. So each of these groups within this population of sharks is interacting with their habitat and their surroundings differently. And so patterns like that may be helpful in being able to predict infection intensities or parasite burden, and trying to figure out who the intermediate host is can be really difficult. So we're going to look at this in a meta community framework. Something to take into account is that that blacktips are primarily piscivorous, so they eat fish. They don't often eat things that are not fish, but occasionally we've found cephalopods and crustaceans in the stomach, and I've read about them eating rays and smaller sharks before as well. It's not very common, but if you think about sharks, they obviously can't touch their food, so juveniles are going to go around and bite everything, and that's how they're going to figure out what is and what isn't food. But that also will expose them to a really wide range of intermediate hosts. All right, so at the among host scale, which is what we see up top, we've got 5 hosts up there. Exposure is going to be determined by a small handful of things: the host traits, the host ecology and regional conditions, and then also the parasite's ability to disperse across habitats. And because, like I said, we don't know much about those intermediate hosts, we don't know a whole lot about parasite dispersal, which is the reason why that meta-community framework is going to be a better way for looking at this system. And so looking at this in terms of a meta-community, each individual shark is one community. So we've got one that hosts 1 parasite, one that hosts 3, one that doesn't have any. But all of these sharks together are what makes the broader meta-community. And so we can look at this within host scale as well. And in that part of the project, we're hoping to figure out where parasites are ending up in the intestine. So we'll be doing distribution among hosts, but then also distribution within host as well. And those within host patterns tend to be defined by the gut conditions and the interactions that are taking place between worms. All right, so I'm going to introduce the idea of filters. I like to think of filters as like hurdles in track. So the worm is going to have to overcome several challenges, several hurdles to be able to successfully thrive inside of a definitive host. And the hurdles that this worm or parasite is going to end up facing while outside of the body are different from what they experience inside the body. So filters to dispersal are going to include exposure, which references which parasites a host encounters, and then also external colonization, which is just whether the worm can colonize that host. Can they get into it successfully? But colonization can also be looked at on the internal level as a filter to persistence. And this is because the internal environment is going to pose its own obstacles to the worm. So the size, the condition of the gut, what's moving through it, those things are all going to determine if that worm can be successful. And then once a worm finally attaches, the last hurdle that it's trying to overcome is the ability to coexist. All right, so as mentioned, external factors are what is going to impact dispersal. Internal is going to impact persistence, and then colonization is the process that connects those 2 scales to one another. And so from habitat to host back to microhabitat, we see that community assembly is something that unfolds in a hierarchical manner. All right, so the first section that I'm going to end up going through after the background section is my among host scale questions. That's the first chapter of my thesis. And so the 2 I'm going to cover today ask about host traits and how that impacts parasite load, as well as geographic region. Pulling back to the question about host traits, some predictions that I have had moving into this were that larger, more mature sharks were going to harbor more parasites. And that's because they have had more cumulative exposure. So they've been living longer, they've had to consume more food, more fish. And if they're larger, they're obviously going to also need to consume more to support themselves. So they are going to end up exposed to these parasites a lot more than we can say of a juvenile. And then I also expected to see variation in parasite load regionally as well, just because the way the hosts interact with their habitats, what intermediate hosts might be available, and what environmental conditions might be going on. Looking at my second chapter looks at the within host scale. And this, like I said, is going to end up looking at spatial distribution specifically. And we find that all of our parasites localize in the intestine. And something, if you did not know about sharks already, is that they have a scroll intestine. So it's not long like ours. Instead, it rolls kind of like a paper towel roll, and that's how you get that increased surface area for nutrient absorption. And so the parasites that are going to be distributed in here, that's going to reflect what nutrients are available, what the condition of the gut, interspecific competition, and then host characteristics. I'd like to show you this figure, which I've chopped up into a bunch of pieces. So this is not the whole figure. What I want you to take from this is on our x-axis, we are looking at a parasite species that was found from another group of scientists that were working on sharks and doing dissections. And they found that this worm is distributed across mostly all of the intestine, but mainly in the central portion. This was another worm that they would have found in the same host. Well, another population of worms, that is. And we see that still we're localizing towards the middle and kind of using the full distribution, but we're more posterior than that first species was. Finally, we have one that localizes directly in the middle and two that look like they either were outcompeted or could not find suitable habitat within the intestine. And so this made me think that when there are few worms present, we'll probably see them all aggregated in that center area. That seems to be the optimal zone for nutrient absorption for them. And when there's many worms, I anticipate seeing more niche partitioning, like we're starting to see up here where they're taking different parts of the habitat. And so ultimately, we're looking at these two different scales that complement each other really well. We're looking among and within hosts at distributions on two different levels. So the environment, host traits, and parasite dispersal are going to be what shapes what sharks we find these parasites in. And then spatial organization and interactions are going to tell us something about coexistence, hopefully. All right, so together they are able to answer a lot of questions, not all of them, unfortunately, but quite a few. And some are, how do host traits and ecological context shape parasite community structure, and what factors contribute to determining spatial organization in blacktips? There are broader implications for this work. Understanding species distributions is really great for when you're trying to protect a endangered species, or if you're working with invasive species. And then these are two awesome worms. But that's all I have for background. It's pretty cut and dry. The first chapter, as I mentioned, is looking at among host patterns, so we can get started with that. All of our hosts were collected by our very own Grubs Lab. They participate in CMAP, Gulfspan, and Big Bend community surveys. So that would have been how I acquired my samples, is through any mortality that occurred. And my sampling would have taken place in 2024 between May and September. That's the time of the year that we see blacktips in this area. So we know that we can at least sample them. And after sampling, because I actually did not pick the sites since they were part of the community surveys, I had some students map them, and we saw that they can be broken into these three different regions. So there's northern sites, central, and then a couple of southern sites as well. And across all of those sites, we were able to collect 32 host individuals. 21 of them were female and 11 were males. Across both male and female, juvenile and mature sharks, the total length ended up spanning from 57 centimeters to 161. So they're not a very big shark. They are usually designated as mesopredators instead of apex predators. And I have to give a shout out to Rollo, because Rollo knows how geographically challenged I am and has made every map that's going to show up in this presentation. Okay, so one of the metrics that we're going to be using moving forward is infection intensity. This is a really easy metric to follow. It is simply how many worms we see in the individual. So you see on that left side, there are 6 worms present. That means our infection intensity is 6. On the right side, there's 4 worms present, so our infection intensity is 4. When we look at those sharks from earlier, you would have 1, 0, 3, 1, and 2. Hopefully that's pretty straightforward. And I hope to use this metric to determine which factors are influencing parasite communities. So all of our sharks ended up being infected. There weren't any individuals with 0 parasites, which is kind of crazy. And we had a very wide range of infection intensities, with some sharks as low as 4 worms found in the intestine, and then others we found 800 plus. So there was big range. And so I ended up taking Shannon diversity here, which is a measure of how evenly parasite counts are distributed across hosts. And I got a 2.65 out of 3.47, which suggests to me that parasite load is not evenly distributed among hosts, which I think, looking at that range from earlier, 4 to 808, is pretty spot on. And so when I threw that into R, we got this really beautiful distribution here that shows that parasites really are aggregated. So there's many hosts on this left side that have low infection intensity, so few parasites. And then there are a few hosts that have very high infection intensity, so very many parasites. All right, so as you can see, this is strongly right skewed, which made statistics questionable. So I ended up log transforming infection intensity so that we could have a more normal distribution to work with. All right, the host traits that I was concerned with were sex, maturity, and size, because of those differences in ecology and how they interact with those habitats. And then obviously the region as well. And so this will tell us about dispersal and exposure opportunities, but I have to give another little shout out to 2 more students of mine, one in which has already graduated, so I don't think she's here, unfortunately. But the other student they spearheaded doing the literature review for this portion of the project, and were able to learn a lot about blacktip ecology to inform those hypotheses. Before looking at infection intensity itself, in their literature review, we found that blacktip sharks typically exhibit sexual dimorphism. So this means that females are going to typically be larger than males. And so I wanted to see if that was represented well in my sample. And it looks like there wasn't really a difference in size. But when we're looking at maturity level, there was. And I think that that makes sense. A mature shark is obviously going to be much larger than a juvenile shark. But what that does tell us is that we might have to look out for some confounding between size and maturity. Looking at intensity specifically, we saw that sex was not a significant predictor of intensity. And then maturity, we saw that there is a significant difference. And so this might be something driving exposure and parasite burden. Because of the fine scale differences that I mentioned earlier in habitat use, I wanted to look at these classes a little closer. So I combined sex and maturity to give me 4 classes. So I'd have like juvenile female, juvenile male, mature female, for example. And I first ran a one-way ANOVA just to see if there was any difference between these groups at all. And it was marginally insignificant, but close enough that I decided to do some follow-up analyses. So I followed that up with a two-way ANOVA, which was testing sex maturity and their interaction, and we did not have a significant interaction effect, which offers some internal validity to those earlier conclusions. And so these trends suggest that mature sharks of both sexes, that is, have higher parasite loads than juveniles. But we see a fair amount of variation within each of those classes, with the exception of the juvenile males. And so we're thinking now that infection is likely going to be influenced by other factors like region or diet. The only one that I hadn't tested yet would have been size. And I started with a Poisson distribution, given that I have parasite count data. And when I put that model together, there was a lot of overdispersion, which suggests that it was not a good fit. So we moved over to using a negative binomial GLM. And this was a much better fit, showing that as hosts become larger, they do in fact have more parasites. All right. We also were able to see differences across those sampling regions. So the central region was not significantly different from either the north or the south, but the north and the south were significantly different from one another. And so this could end up reflecting what intermediate hosts were available and what exposure opportunities looked like for these individuals. So if we're going to summarize all of the traits that I've just tried to pull together, when making the final model, once size was integrated, we did not see a significant effect of maturity or region anymore. So this was the final model that we ended up going with for best explaining infection intensities. And essentially it just indicates that cumulative exposure and habitat range is going to be wider for those mature sharks. All right, so to recap what we've found here, parasites are highly aggregated in some individuals in the population. So we usually see those very low infections, but occasionally see those very heavy infections. Host size is the strongest driver we have so far of infection intensity, but maturity and region could contribute some variation, just not nearly as much as size. And so we've only looked at host traits and regional variation. I haven't looked at parasite dispersal yet. And that would be because we don't know about that intermediate host. So that's why I haven't been able to gauge dispersal. Moving towards our next part of the talk, infection intensity can also be shaped by those characteristics that I was talking about that occur inside of the shark. And so this is going to mean the intestinal environment or interactions with other species are going to be what tell us what species can persist and where in the intestine they can persist. So we'll be moving from that meta-community level down to one individual host, which serves as one single community. All right, so we're moving towards chapter 2, which goes over within host patterns as mentioned. And so we'll be zooming in to the intestine of the shark shown here. Each of those intestines functions like a closed system, a mini ecosystem of a sort, and it can be totally censused, as I mentioned earlier. So we can account for all worms that are in that organ. And so what I would like to know is whether the same factors that were influencing infection intensity outside of the host are impacting colonization within the host. And I have to give another shout out, of course, to my undergrads. When I was at the marine lab, we had a pretty foul job some days doing the intestinal dissections. And I appreciate that on the days when my stomach couldn't handle it, my students never gave up. They really were the muscle. All right. Back to that within host ecology scale though. So parasite placement is going to end up being impacted by host characteristics, habitat suitability, and those interactions taking place as mentioned. And how did we gauge spatial organization within the intestine? This was actually, it took a lot of thought, but it actually wasn't that complicated once we figured it out. So this square, imagine that to be the shark's intestine. When you unscroll that scroll valve, it's roughly rectangular. So we can use surface area as, well, to our advantage. So I ended up where I made 5 centimeter cross sections, which is what we've got going from anterior to posterior here. So cross section 1 is 5 centimeters wide, and it's the anterior-most piece. And that's the case for all sharks that we did our dissections for. And then the posterior-most part of the gut is going to be cross section N, whatever that last one is. And then under microscopes, we were able to extract parasites from each of these sections. And because we had set it up that way, we were able to log the total parasite, not only per host, but per intestinal cross-section. All right, so a couple different statistics that we're going to use moving forward. I guess this one's not really a stat actually, but relative intestinal position. We were trying to put the intestine on a scale from 0 to 1 so that we can standardize placement or relative placement within hosts that have different sized intestines. And then after doing this, we came up with weighted mean intestinal positions for each individual host. And so this would have been the average relative position of all of the parasites weighted by the parasite count per section. And so this is going to tell us where the highest concentration of parasites is in the intestine. This is one of my favorite figures. It shows all 32 of the hosts. So they are designated as CL and then the host number. CL is Carcharinus lymbatus, scientific name. And again, we're moving left to right from anterior to posterior. And what we see is that in most cases, we're centrally located. Parasites are obviously not uniformly distributed. That section of 3 hosts shows a really good representation of that central clustering, but we also saw some really funky distributions here too, some that showed completely opposite trends of what we saw in most cases. And then we also had a few that showed bimodal peaks, which made me excited because I had hoped that that might mean there were interactions driving that. I overlaid all of those curves from each of the 32 hosts into one curve to get a general idea of where the worms were. And they did show up in that central portion. When I calculated the mean weighted position, it came out to 0.47. So if 0.5 is the midpoint of our gut, we're pretty close to central. And so these patterns are going to be shaped by habitat suitability, gut physiology, host characteristics, and then those interactions that are taking place within the parasite community. And so I wanted to revisit host traits and see if they influenced parasite distribution in the intestine. And I kept these as part of my questions and hypotheses because I did anticipate that the gut might have differences, whether it's chemical differences or morphological differences between these groups. And so by intestinal characteristics, some of the things that I'm referring to are availability of nutrients, pH, flow rate of organic matter moving through the organ, mucosal thickness, and enzymatic activity. So we've got our rectangular intestine schematic here again. And what we know from the literature is that the gut is partitioned into about 3 different sections functionally. And so in the anteriormost portion, we have very low pH from stomach acid. We have high levels of enzymatic activity from digestion. And then we have thick mucosa because the intestinal lining needs to be protected from that low pH. This is not going to be a good place for a parasite to thrive. Now, in the center, food has been digested more, so there's more opportunity to uptake nutrients, and the flow rate is also slower. So they are collecting as much food as they possibly can if they're in that region. Region in the back, nutrients are very sparse, and worms are very easily expelled if they colonize back here. So that's not always a safe option. Now, how did I think that was going to impact the gut specifically? For the most part, I was anticipating changes with maturity and size. Less sex, but still wanted to throw it in there to try and test it out. I anticipated that as sharks are older and larger and have consumed more food, that those digestive enzymes are going to be pulled from the anterior portion closer to the posterior portion as that food is continuously moving through. So I think that that anterior portion of non-optimal habitat is going to grow. I think that our parasite distributions are going to shift backwards, and then that last section is going to be pretty tight. Looking between sexes at mean relative position, let me get you accustomed to this figure. So now we have the gut on the y-axis instead of the x-axis. So this shark here is supposed to help orient you with anterior down towards 0 and then posterior is up towards 1. And we see no difference between the sexes here. All right. Well, there was a difference between maturity classes, and it looks at least kind of hopefully like my hypothesis to a degree, because these juveniles have this tighter range of space that these parasites are taking up, and then mature individuals have a wider range and more parasites in a slightly posterior shifted region. Next, we tested host size, and this was also a significant positive relationship, telling us that as sharks increase in size, we are seeing the concentration of parasites shifting ever so slightly further towards the posterior end of the intestine. And so this is generally what we got when we were looking at the worms. We see that they're centrally clustered in the intestinal habitat, but I wanted to look closer at some of the fine scale organization. So how are those parasites spread and distributed just within that small area that they occur? Okay. So going back to low and high intensity real quick, low intensity is going to mean we don't have very many worms. So I would assume that all of our worms are going to cluster in that optimal zone. As we get more and more worms, that part of the intestine is going to fill up and there's going to be less available habitat. And my prediction was then that we would see more even distribution across the intestine because more of it would be used as habitat once it was filling up. To figure this out, we pulled Shannon diversity back into the mix to measure the diversity of parasites. This time, instead of across host and across intestinal sections, And we were able to standardize Shannon diversity, which you would usually standardize by parasite richness, but we did not. We standardized it by the number of intestinal sections with parasites. And that's because this was supposed to be a proxy for spatial richness. And so if we have high evenness, this is going to mean that our parasites are spread pretty uniformly from front to back. And if we have low evenness, that'll indicate clusters. This figure really threw me for a loop when I first saw it. On the left is infection intensity by spatial evenness. And so there's a slight increase there, but really not much happening at all. But you'll notice that most of the points are close to that 0 to 200 or 0 to 400 range in infection intensity. So I really think that it was these outliers pulling that line to make that trend. So I pulled log infection intensity back into the mix, and the line goes a different direction. That stumped me, but regardless, they are pretty stable throughout. So evenness was stable from front to back. And so if spatial spread is independent of parasite load, this tells me that evenness is going to be more influenced by the intestinal characteristics than by total load and those interactions. To wrap up everything from this section, quick summary would be that parasites are aggregated, host size and maturity broaden those distributions and shift them posteriorly. Spatial evenness remains stable regardless of infection intensity. And then interactions among the parasites inhabiting the intestinal community have not yet been investigated. All right. Moving into the next portion of the talk, exploring parasite richness. Richness is similar to intensity, but not quite. So this is going to be the distinct number of species that you find. And in this case, we're calling them morphotypes because I haven't yet identified all the way down to the lowest taxonomic class for all of them. But to gauge richness, we've got two different colored worms on the left. So that is going to represent two different species of worm, which means we've got a richness value of 2. On the right side, we have 5 different colored worms, which would indicate 5 different species, so a richness value of 5. Now, what we're moving into is still in progress, but I figured I can't talk about worms and not show you some exciting pictures of worms. So that's what we've got coming up. You'll get to meet the monsters. So I have been able to do identifications for 5 of the hosts so far. And in those 5 hosts, we have found 36 different morphotypes. This would be the first 13. And I mean, I'm not going to go through all of them, but maybe it's worth taking a look at numbers 3, 6, and 7, because those were highly abundant compared to the other taxa that were in there. We have another 12 on this page, and this is mostly our species that have a leaf-like head structure. So it's almost like they have petals that they use to suction and attach. The other group that we found in here to be pretty prominent would be Onco1, which refers to parasites that had fangs. Finally, this is our last group, and you'll notice that we mostly see that TRP code. So we're tripanoranx all around, and tripanoranx just means that these are going to be our parasites that have tentacles that they can expel. And so that's what you're seeing in most of these guys with it is coiling. That's going to be their muscle sheaths and their tentacles. Now, as I mentioned, The analysis is still in process, but I wanted to give you a little bit of something. So for the first host, CL01, we had 4 parasites and 1 of each different designation. So they were split evenly. In the second host, we jumped to 14 different morphotypes that I was able to find in there. In CL03, we jumped up to 23. And then for Shark 4, we fell back down to 5. I do recall that I think this was a juvenile, so that might be why. And then the last host I did really stole the cake. 28 whole different morphotypes was kind of crazy to me. And you can see there's definitely some front runners and then others that are not doing so well in there, or don't have a big hold on their population. So richness ended up varying from 4 to 28 morphotypes across these 5 individuals. And once I move into the analysis, I'm again going to pull those host traits back around and see if those influence diversity. And I would anticipate that larger sharks, as they had more parasites, are also going to have more taxa present, reflecting increased exposure over time. However, it's important to note that I'm going to have to try and disentangle the relationship between intensity and richness, meaning this large shark on the right, does it have more taxa because it has more worms? So I have to work on that. And then I'm also going to be revisiting the questions regarding region. And this is because parasites can get to their host in many different ways. So in this life cycle, we would have 2 intermediate hosts, the snail and the fish, and the definitive host would end up being the shark. But it doesn't always have to follow that. Sometimes we'll have chains of longer than just 1 to 2 intermediate hosts. And then finally, other parasites might move by a whole different transmission pathway. And so this will increase diversity is what my hypotheses are. Okay, so I'm anticipating that increased richness will increase the degree of niche partitioning and spatial organization here in the gut. I think that this is going to end up giving us a very patchy distribution in the end. And I'm hoping to look further into how these parasite taxa are interacting with each other within the host. So I anticipate that some morphotypes are going to show up consistently together or consistently in most hosts. And to me, that would suggest that they can handle the same conditions in the gut, but also that maybe there is a shared pathway of transmission. Maybe they're both moving through the same intermediate host to get to that definitive host. Others might end up segregating, indicating that they are competing and are not compatible with one another. So I intend to look at which taxa are co-occurring more or less frequently than others. For the analysis, I intend on using a non-metric multidimensional scaling analysis, NMDS. And so this is going to help us visualize the differences in parasite community composition across hosts. And so when looking at this figure, what I would like you to know is that each individual point refers to one individual host. And so how close they are to each other on the figure indicates how similar or dissimilar they are from one another. This is the face I made when I realized I'd have to make one of those figures. I'm going to follow that up with a permanova, and that's going to test whether the community composition is varying with host traits, regional variation, intensity, richness, or any of those other factors that I'm trying to pull in. And so this will end up telling us what's causing the composition of these communities to be less similar or more similar to one another. reaching the end of my presentation, I'd like to just go over what analyses are ongoing and upcoming. I'm currently assessing sampling completeness using species accumulation and rarefaction curves. So essentially trying to figure out if we can subsample these worms, since there is a total of 4,000 of them, to be determined. I'm going to have to finalize those parasite identifications so that I can carry out that richness analysis and the analysis concerning community composition. But I'm excited that once I have these, this data and can run these stats, I hopefully can start to link these two scales of ecology a little bit better. And so I do have some diet data from these black tips, but unfortunately, most of the stomachs were empty. So I really don't think it's going to be all too fruitful of an investigation for me. But I'm hoping to look at what prey items are showing up in their stomachs and see if this can help us by providing any hints for these pathways, given that not much is known about the intermediate host, and that is the key to understanding parasite dispersal, it seems. So I'm shouting out these students with a really crazy picture of us because at the end of all of our shifts, I think that our sanity was not evenly distributed. And they at least could make me laugh so that I'd want to come back the next day. What I'm shouting them out for, though, are a couple different hypotheses they've come up with as well. My students have pointed out to me that after we noticed there was a difference between the north and the south sampling sites as far as infection intensity goes, that there are some potential contributors to why these regions might be different from one another. And one of those is that those northern regions are near the Appalachian Bay. And so we might see differences in salinity or productivity, and we'll find lower salinity up towards the Appalachian Bay since it's an estuary. So if anything, I hope that this work can demonstrate that parasite communities can reveal ecological processes at multiple scales, but I also hope that it can help offer insights into understanding the structure of biodiversity in ecosystems and in individual organisms, understanding how host life history and movement influences exposure and infection risk as well as parasite dispersal. And then also, I think we can understand some broader implications of symbiotic communities for marine food web dynamics. I think that in many cases, we don't think about what's inside. So ultimately, among hosts, we see host traits and regional variation driving this. Within host, we see very consistent patterns occupying that central region, but also some effect of host maturity and size. And then intensity having little to no effect. And if you did not take anything else away from this presentation, I hope that you can at least walk away with an appreciation for parasites, because parasite communities may be hidden. We forget about them, we don't see them, but they tell a powerful story. They tell us a lot about evolution and ecology. With that, I've got to do some quick acknowledgements. I don't know who to acknowledge for that awesome picture. I've got 1 million students and somebody drew it and I can't figure out who it was. I swear I've asked every one of them. But yeah, I have to thank my students. They're really awesome. I wouldn't be able to get through 4,000 worms without them. I've got quite a few of them, and this one doesn't even incorporate the ones that started this week. So I've also got to thank my advisors, Tom and Tara, Dean for helping supply specimens, and Scott as well, and then all of my grad students and friends and funders. It has, the parasite load
and lethality in juvenile sharks? I mean, is there a, I know there, you know, this is an early, you know, one for survey, but you know if there is a parasite load in juvenile or small shark that would be used? I mean, I can see that that would be difficult in the census because we don't know if they're acquiring one parasite is there, but it don't help. But, you know, but I'm just wondering if purely acquisition over time, or if you're just not seeing high loads in juveniles.
I think it's definitely worth looking into, and I think it could be worthwhile to compare among other sharks that are that size. So the S. tiburo, does anybody remember the common name of that? I can't remember off the top of my head. Bonnethead. They're smaller and might be closer in size to like a juvenile. It might be worth looking at that. Could I have you repeat the beginning part of your question though?
Yeah. If it's what's known about, you know, lethality, just in terms of at a high parasite load, they might be leaching out at risk of nutrients, but, you know.
Yeah.
I mean, mammals usually be well deficiency, but.
Yeah, when they have low intensity infections, it usually doesn't really bother the host all too much because they're not taking too many nutrients. And it's only once we get to those really, really high infections, say 400 and up, that we see that there are noticeable impacts on the health.
How mobile are the worms once they're in the intestine? When I'm wondering about whether your distribution represents the worms settling when they first get to the intestine, you'll see drop off of worms dying where they started from, or are they moving around and finding the best path?
So from what I understand, once they make it into the intestine, they will kind of ride their way through until they reach that optimal zone and take hold, but they can be dislodged and move further backwards. I don't know how mobile they are once they're in there outside of that. But I don't think that they move very much.
It looks like many of these worms or parasites are microscopic, hard to follow, which makes me think that larvae and media hosts even harder to follow. I'm wondering if you thought about like DNA barcoding for those barcode probes to look for presence of parasites and different hosts without having to do dissection, searching for microscopic level parasite muscle.
Yeah, and there has been a lot of work on figuring out metabarcoding and figuring out their genome structure and whatnot. I didn't do any of that in this project just due to funding and time restraints. But with such a wide collection, I hope to at least be able to pass it on to some of the others in my field that are trying to work on connecting those pathways.
Anthony has that building barcodes for 18s. I don't know how
Yeah.
good just standard 18s primers are
For them.
for them,
Yeah.
but it's just a yeah.
I'll have to look. I've definitely seen a couple. I don't know. I don't think it was 18S that I saw, but I don't remember off the top of my head either.
Something I'm with you on. It's like, could there be a bias in the fact that is it really stressed out individuals how well for they might go and fight? They can't have those things, so there's more like this cost because it's stressed out. Is there anything else that
It's definitely possible that we have like hosts that are mainly in poor condition that were caught. And so one of the things that we had wanted to look at originally was like take some blood samples, look at hormones and try and get an idea of the health and the stress of the animal. But unfortunately, since by the time I got them, they were dead, those levels had evened out and we can't get that kind of data. But I definitely think that given that some of those individuals had very high parasite loads and then just kept acquiring, that it is a sensible hypothesis at least.
Stomach acid, malnutrition produces these higher features, not much acidity. And, you know,
Hmm.
the pH level
Yeah.
might have got messed up by the time you got, but I wondered, does that include in sharks if they're nourished, do they, I mean, if you were to take the pH measure, and add.
I am not totally sure, but I mean, I imagine that if you see it in mammals, maybe there's a same evolutionary. I don't know. I really don't. But thanks for bringing that up. It's a good question.
First of all, countless days of brain not where I saw you. Countless hours with your head in a shark.
Yep. 8 a.m. to 8 p.m.
And I know, but
Mm.
the placement based on those morphotypes, like you said, the thicker walls and some different morphotypes.
To that end. I haven't noticed anything with the spatial organization, but I have noticed two parasites that are occurring pretty regularly in all of these host individuals. So I am at least able to figure out who's a generalist maybe, but beyond that, don't have a whole lot.
Right, well, do you want me to thank you?